Okay, in today's video lecture, basically I will going to talk about the lock and key model and the induced fit model. Both these two models are very much important to know that how an enzyme will bind with its substrate or to know about the enzyme substrate specificity. So let us begin today's topic. The first model, which is the lock and key model. This model is proposed by Emil Fischer in the year of 1890. In his model, in his lock and key model, the Emil Fischer described that the process through which a lock will be opened or a lock will be unlocked by using its particular key. Similarly, an enzyme will bind that type of substrate which have the structurally similar configuration with the binding site of the enzyme. So, if we take an example, let's suppose this is our lock. This is our lock, and the hole through which we enter the key to unlock or to open this lock is known as the key binding site of this lock. Now, let's assume that this lock here, the lock is acting as an enzyme. The lock is acting as an enzyme, and the key binding site of this lock is known as the substrate binding site of this enzyme. If you guys remember, in my video lecture I have discussed that the site where the substrate will come and bind with this enzyme to form the enzyme substrate complex is known as the substrate binding site or the active site. Similarly, in this case, where the substrate will come and bind with this enzyme, this site also known as the substrate binding site or the active site. As we assume this lock as an enzyme and the whole of this lock where the key will enter to open this lock is known as the key binding site of this lock. That's why the key binding site of this lock is also known as the active site of this enzyme, if you will consider this lock as an enzyme. Now, let's assume this is the key. This is the key to open this and to open this lock. This is the particular key to open this particular lock. If you use different key, we will not able to open this lock. So this is the one and only particular key to open this lock. Now, when we enter this key by using its key binding site, at that time this formation is known as the lock and key complex. And if we consider this lock as an enzyme, and if the key is considered as an substrate, at that time this formation is known as the enzyme substrate complex. So the lock is similar to the enzyme, the key is similar to the substrate, and the lock and key complex is similar to the enzyme substrate complex. Now, this complex structure is known as the lock and key complex or the enzyme substrate complex. Both are similar. Now, look at this picture. This is the key which is acting as a substrate, and this is the lock which is acting as an enzyme. Besides, this is the enzyme and this is the substrate, and this is the active site of this enzyme. Now, when the key will come and bind with the key binding site of this lock, they will form the lock and key complex. Similarly, when the substrate will come and bind with the active site of this enzyme, they will form the enzyme substrate complex. Now look at here. I have mentioned here that the lock is acting as an enzyme and the key is acting as a substrate, and the lock and key complex which is formed here is similar to the enzyme substrate complex. So this is what about the sense of lock and key model which is proposed by Emil Fischer in the year of 1890. Now, in his model, he also mentioned that the active site is a rigid in structure. That means, let's suppose this is the enzyme, the red one is the enzyme, and this is the active site of this enzyme. Now, you can see the structural configuration of this active site. Now, the green one The green one is the substrate of this enzyme. Now, if you look at the structural configuration of the active site of this enzyme and the structural configuration of the substrate, the structural configuration of the substrate is exactly similar. There is no difference between the structural configuration of active site of an enzyme and the structural configuration of substrate. Now, due to this similarity, the substrate will easily bind with the active site of this enzyme and they will easily form the enzyme substrate complex according to the lock and key model which is proposed by Emil Fischer. Now, here in this picture, the substrate will bind with the active site of this enzyme and they will form the enzyme substrate complex. And after some time, due to the enzymatic reaction, the enzyme will convert this substrate into specific product or a particular product. But the characteristics of this enzyme or the active site of this enzyme remains unchanged. So after the enzymatic reaction, the enzyme will be free and the substrate will be converted into a specific product, which is the blue one. Now, if you look at this diagram, you can see that the structural configuration of this substrate is similar to the structural configuration of this product and also similar to the structural configuration of the active site of this enzyme. This characteristic is known as the template. So what is the template? Let's assume you used a round shaped glass to make an ice cream. Now, when the ice cream will be formed and when you will take it out, the ice cream from this round shaped glass, you can see that the shape of this ice cream is similar to the shape of this glass, which was the round. Similarly, the substrate which will bind with this enzyme, the structural configuration of this substrate will be similar to the structural configuration of this active site, and the product which will become after the enzymatic reaction or after the formation of enzyme substrate complex will be similar in structural configuration. It is similar to the structural configuration of the active site of this enzyme. That means the structural configuration of the enzyme and the structural configuration of the active site of an enzyme and the structural configuration of the substrate and also the structural configuration of a product is quite similar. This is known as the template. So this is what about the sense of lock and key model. Now moving to the next slide. In the next slide, there is the induced fit model. This model is proposed by Daniel Ecosland. The induced fit model is nothing but the modification of lock and key model. This is the updated version of lock and key model. If you guys remember, in the lock and key model, I have said that the active site of an enzyme is rigid. That means the active site of an enzyme will bind with that type of substrate which have the structurally similar configuration of this active site. The active site of an enzyme never changed. The active site of an enzyme will never change. But in induced fit model, okay, before this I want to mention one thing, that the lock and key model, was rejected. So why the lock and key model was rejected? Just because there are some molecules which can easily bind with the active site of an enzyme because they have the structural similarity with the active site. Let's suppose this is our lock, and to open this lock I have to use its particular key, and we can easily open this lock and we will get our product. But sometimes you can see that the lock, you can also use another key to open this lock, but you will see that the key will easily enter into the lock, but they will not able to open this lock, but they will easily enter into the lock. Let's suppose this is the another key, but if we enter this key into the lock, it will easily attached with the lock, but it will not able to open this lock. Similarly, there are some molecules which have the structurally similar configuration with the active site of an enzyme, and they can easily bind with this active site, but they will not form any kind of product. So that's why the lock and key model was rejected. Now, in the year of 1958, the Daniel E. Kostler, he proposed another model, which is the modification version of lock and key model. This is known as the induced fit model. In his induced fit model, he said that the active side of an enzyme is not rigid. The active side of an enzyme is not rigid. It is flexible in nature. So, in induced fit model, the Daniel E. Kostler proposed that the active side is flexible. That means the active side of an enzyme will bind with the substrate which may have not be the similar structure and configuration of the active site, but have some little bit similar structure and configuration of this active site of the enzyme. Now, if you look at this picture, this is the active site of this enzyme, and this is the structural configuration of this active site. And let's suppose this is our substrate. This is the structural configuration of this substrate. Now, there are huge difference between the structural configuration of substrate and the structural configuration of this active site. But due to the flexibility nature of active site, the substrate will easily bind with this enzyme, and after the enzymatic reaction or after the formation of enzyme substrate complex, the enzyme will easily convert this substrate into specific product, which is the green one. So this is what about the sense of induced fit model. So I think all this data provided by me will be very much important to improve your study and to improve your knowledge about the enzymology. So I think this much is sufficient for today's class. So thank you for watching my video.